Okay, this lecture will be about the digestive system infections. This comes to us from chapter 25 of our textbook. As we've done in the past for our survey of the infections of the human body, we're going to start by talking about the anatomy and function of the digestive tract, some of its natural defenses, and its normal flora. Then we'll move on to talking about the bacterial diseases associated with this part of the body, the viral diseases, protozoan infections, and finally the helminthic or parasite sort of infections. Okay, so your anatomy and function of the digestive tract includes all of the tissues that are part of the mouth, as well as some of the accessory tissues like the teeth, the parotid gland, the salivary gland that'll become important in a little bit. Also, the lower digestive tract is going to include things like the stomach, the intestines, and their accessory organs, including things like the liver and the gallbladder and the pancreas, etc. The purpose of this entire system is to digest food. Absorption is going to pass nutrients from the small intestine into the blood or lymph for distribution throughout the body to feed all of your cells. So digestive infections are very, very common. In fact, they're second only to respiratory infections in terms of the most common illnesses for humans. Annually, we see about 76 million cases for foodborne disease, and that's probably an underreport. A lot of people don't go seek treatment when they have a GI illness, as long as it resolves within a day or so. But it does account for quite a bit of missed time from work, reduced productivity, and on a more serious level, 5,000 deaths per year. It is usually due to, it is almost always exclusively due to the consumption of contaminated food or water. So that would be a vehicle transmission, something that you ingest. So therefore, it is most commonly due to the fecal-oral route of transmission. So you are colonized from birth, from the moment that you're born until the moment you die, and even thereafter, microbes will help to digest your body. So from the moment that you exit your mother's uterus, you are colonized by bacteria. You will have less organism in the stomach and the small intestine. Can you imagine why that would be the case? It has to do with the low pH present in both of those environments. However, if organisms are able to stow away inside of a food particle, they will make it all the way to the colon or the large intestine, where there is an enormous microbial presence. In fact, there's about 100 billion bacteria per gram of your fecal matter. So a lot, a lot. They tend to be mostly anaerobes and facultative anaerobes. Why would that be the case? Well, this is pretty far removed from any source of oxygen. It's not in the mouth. It's not close to the skin where there's plenty of oxygen available. And it's not really attached to the bloodstream where there would be an oxygen supply coming from that. No, instead, they're kind of trapped internally inside your body. And so therefore, they need to be able to handle low oxygen conditions. But they do form a commensal relationship with you. They tend to break down some of your foods for you. Think of the lactose in milk. We know that many of our gram-negative Enterobacteriaceae organisms are lactose fermenters, so they will help with that. They also make useful vitamins for you, including vitamin K. And we kind of already gave it away. How did they get here? Usually it's from stowing away inside of a food particle that didn't get broken up entirely in the stomach or small intestine. Some of them may also be acid resistant. Spores, for example, could potentially be an issue, although not super often. Even though it's internal to the body, your GI tract is technically considered an external body surface. It's protected from your blood, your lymph, and the parts of you that are inside. That's actually a mucosal lining and epithelial tissue that surrounds your GI tract. And so because of that, it is still considered an external body surface. All right, so defense-wise, about 80% of your immune system is stationed inside the GI tract, and especially the small intestine. And there you'll find these special tissues called the gut-associated lymphoid tissue, or GALT. And basically, it's a great big sort of like military base, if we were to use that analogy, of all of the things that make up your immune system. They are there because this is a huge site of interaction with the outside world. You're also protected to a certain extent by the low pH of your stomach and small intestine. That will be inhibitory to growth and even bactericidal for a lot of things. But some things will survive. And then you also have these things called Paneth cells, which are granule-containing phagocytes. So basically, those granules are something that once the organism is phagocytosed or swallowed, the granules will release onto those target organisms and degrade them. It will include things like defensins, which are a special host defense antimicrobial peptide, and lysozyme, which will help to lyse those cells. Lastly, your stomach and intestinal lining produces mucus, and this mucus helps, just like it does in the lungs, to trap organisms. In addition, your intestinal lining will slough off and be regenerated every 2 to 6 days. So if something does happen to get in there, they're regularly dividing cells so that they can be replaced. So we'll start in the mouth. And really the things that are most important here are most common are going to be things like dental caries or cavities, gingivitis, and periodontitis. Now, I'm not going to go through these very much because those are things that are not really within the medical side of things. They're really more of a dental concern. They would need to go to a dentist in order to seek treatment for that. But one thing you do want to try to keep an eye out for is this thing called acute necrotizing ulcerative gingivitis, which is also known as Vincent's disease or trench mouth. Its symptoms include really painful chewing, and its hallmark symptom is super foul breath, which is called halitosis. And it will form sometimes a gray-yellow pseudomembrane on the gum line. So this is a mild case of the illness. And over here, we're seeing that the gums are actually receding. We've got this swelling right here, and this gray pseudomembrane is forming. So that's a more advanced or acute case. The etiology of this includes an organism called Prevotella intermedia and other organisms that can do this. To treat this, you're going to be using oxidizing agents, debridement, so removing tissue, maybe pulling teeth, metronidazole, and antibiotics. Most of the organisms that are causing this are going to be anaerobes because the mouth is closed for so often. And so that's why things like oxidizing agents will be important. So for your lower digestive system, I want to make sure that we differentiate between infections versus intoxications. So with infections, this is a pathogen-driven illness. And the organism can multiply while it's attached to the gut epithelium, or it can cross using these cells called M cells that are part of your mucosal lining in order to access the lymphoid tissues. These are also known as Peyer's patches. And when it does this, it can trigger an immune response. Now, these infections are generally a delayed response from the time of exposure because the pathogen has to multiply before symptoms are observed. And they will usually be accompanied by a fever. Antibiotics are usually not going to be helpful in these situations. Why do you think that might be? Well, sometimes they're self-limiting, so they will take care of things on their own. A lot of them will produce toxins, and that will be sort of a side effect of what they're doing. So that can be potentially an issue. But again, most often, a lot of our GI infections are going to be viral. And so with that, antibiotics are also not going to be helpful. But if you do know that you have salmonella or you have E. coli or you have cholera, there might be antibiotics that we can use to help. So that is what's going on with an infection of the lower GI system. Let's compare that with what's going on with an intoxication. Instead of being driven by the organism, this is illness driven by the presence of a toxin. And sometimes the pathogen is not even present. So examples of this would include Staph aureus, food poisoning, and botulism. Botulism is a super weird one. Because the toxin doesn't just affect the GI tract. In fact, it really doesn't at all. It moves into the nervous system. The GI system is just its point of entry. So with intoxications, it's going to be a much tighter timeline between exposure and illness. So it'll be usually within a few hours of exposure. So if you eat something that was maybe a little bit questionable and you're throwing up 2 or 3 hours later, it's possible that you have a foodborne intoxication. Otherwise, you need to be thinking about what have you eaten in the last 18 to 24 hours, because it's more likely that that might be the case. With intoxications, fever is a lot less common. So with infections, think fever. With intoxications, there would not be a fever. So both infections and intoxications will result, excuse me, in diarrhea, abdominal cramps, nausea, and vomiting. This is a major, major problem in developing countries where sanitation is not up to the same standards as what we have here in the United States and in the developed world. In fact, for kids under the age of 5, there's a 25% mortality that diarrhea will kill them during that early phase of life. Treatment for these kinds of infections is going to be mainly driven by oral rehydration therapies. So this is, drinking plenty of fluids, Pedialyte for the really young, that sort of thing. But you can have an advanced stage of diarrhea called dysentery. And if you played the game Oregon Trail while you were growing up, this is one of the things that would kill people in that game. But dysentery really is severe diarrhea that's accompanied with blood or mucus. So that mucus is advanced shedding or sloughing from the mucosal lining. Blood is obviously a big concern and a bad sign. So gastroenteritis generally is any sort of inflammation of the stomach and intestinal mucosa. This is what we usually refer to as the stomach flu, but it's not actually caused by influenza, even on the viral side. It truly is a gastroenteritis. So we'll start first by talking about staphylococcal food poisoning. This is caused by Staph aureus enterotoxin. It's actually an intoxication, not an infection. So what happens here is that this organism is very resistant to things like osmotic stress, and this allows them to survive well in certain foods like cured ham that have a high salt concentration. They tend to have a really easy route into the food because hands are what's used to prepare a lot of our food, and your hands tend to have Staph aureus on them. The toxin, the enterotoxin that's produced by Staphylococcus aureus, can survive up to 30 minutes of boiling, so it's very heat stable. Now, I want to make sure that we differentiate between enterotoxin and endotoxin because they are in fact different. Endotoxin is that LPS component of gram-negative organisms. So it's released as they divide or they die. Enterotoxin is a toxin that affects the enteric system. So it's going to affect the GI tract. But in this case, for Staphylococcus aureus, it's not an integrated part of their membrane. So understand that difference. So I mentioned ham as a potential high-risk food, but others include things like custards and cream pies, things that have that high sugar or high salt content, prepared by hand usually, and are not going to be cooked further before you eat them. For prevention, you just want to make sure that you provide adequate refrigeration during storage to prevent toxin release. This infection, or not infection, it's an intoxication, is very rarely fatal. The diagnosis will consist of the symptoms as well as the short incubation time because in about 1 to 6 hours after exposure, you would see symptoms, and that short suspense would indicate the presence of a toxin, not necessarily an infection. So next we'll move into dysentery, which I mentioned just a minute ago. Bacillary dysentery means that it's caused by a bacillus. In this case, it's Shigella, Flexneri usually, but other species as well. And the disease itself is called shigellosis. Shigella and its different subspecies are anaerobic gram-negative rods, so they fit well inside the GI tract. They are a very close cousin to E. coli. In fact, for a long time, we thought they were the same organism. It wasn't until we started doing 16S ribosomal sequencing that we truly understood that there was a divergence. They're very similar. Its epidemiology is interesting, though, because unlike E. coli, it has no natural reservoir other than humans. So this isn't something that's going to be in the soil. It survives exclusively in the gut and is present in the fecal matter. So therefore, it's transmitted through a fecal-oral route. Outbreaks are going to be most common in families, daycares, the potluck sort of situation. And this accounts for quite a few cases annually, about 450,000 cases. But ordinarily, under normal conditions, this is mostly going to affect kids under the age of 5 who haven't really developed great hand hygiene practice yet. The symptoms of this are pretty intense. It's over 20 bowel movements per day, abdominal cramps, and fever. To diagnose this, you would do a culture from a rectal swab, which is really fun to do to a kid. Let's talk a little bit about its pathogenesis. So here's what that M cell looks like that we were talking about earlier. So you'll have these ciliated epithelial cells, and then every so often you'll have this M cell that doesn't necessarily have the cilia, but it's there to sort of monitor things. And so Shigella will come in, it'll trigger that membrane ruffling that we saw earlier for Salmonella, and then it will be taken in by the M cell. Now it survives phagocytosis. It actually kills the cell that it was phagocytosed by, including a macrophage. And from there, it can spread through the tight junctions into the neighboring ciliated cells. In this way, it avoids the immune system. And so that's a real concern. If this continues and progresses, killing the cell altogether, this will form an abscess, and so you'll have like an intestinal abscess. The organism will rarely spread into the bloodstream. It's usually caught before it leaves this and gets to the blood or lymph. So treatment for this is going to be using antibiotics and oral rehydration, so picking up those fluids as much as possible. The last thing I want to mention is that shigellosis or Shigella is really well known because many of the species will produce what's called Shiga toxin, which is one of the strongest or most deadly toxins that affects the GI tract. This is especially going to be true in Shigella dysenteriae, which thankfully is the least common in the US. It is found fairly frequently in the developing world. This will trigger a bloody diarrhea. So it's usually a bad sign, and you would, as the patient, would know to seek treatment if it's available. Next, we'll move on to salmonellosis, which is a gastroenteritis caused by salmonella. Now, with this, Salmonella enterica is actually the name of the organism, but there's 2,000 serotypes within that. So you'll hear things like Heidelberg and I used to work with Salmonella, and right now I'm missing a lot of them, but you'll see different serovars or serotypes that are associated with that, but the organism itself is called Salmonella enterica. It is a gram-negative rod and a facultative anaerobe. So again, it's going to enjoy being in the intestinal environment. Now we can break Salmonella enterica down into two different things. You can have the typhoidal Salmonella, which is going to include things like typhoid fever. And then you can have the non-typhoidal version, which is going to be a milder form of typhoid fever. So symptoms for all of these are going to include things like a moderate fever, nausea, abdominal pain, cramps, and diarrhea. There is no treatment antibiotic-wise for this, but we do use oral rehydration therapy. For its pathogenesis, this one has a much longer incubation period than the toxin-mediated illness that we saw with Staph aureus, for example. This one is going to take 12 to 36 hours before you see symptoms. And it's going to follow a very similar pathogenesis to what we saw with Shigella. It's going to enter into those M cells, survive phagocytosis, multiply inside those mucosal cells. That triggers an inflammatory response leading to diarrhea. And occasionally the bacteria will cross the epithelial cell membrane to enter the lymphatic and bloodstreams. So it's going to have really rapid division inside of macrophages. It does well there. It survives phagocytosis. And so when a person is actually ill with this, they'll have 1 billion cells of salmonella per gram of feces during acute illness. And when a person is having diarrhea, that's messy, right? That's a messy process. And so that gets everywhere, and it's hard to keep things clean, which really favors salmonella because it allows it to transmit to others a whole lot easier than it might otherwise. When death occurs due to salmonella, it's usually because of septic shock that would happen. So remember, this organism is able to get into the bloodstream and trigger that massive immune response that will cause the bloodstream to become more leaky, resulting in dangerously low blood pressure. For most cases, though, there will be recovery within a few days, but you as an individual, even though you're feeling better, will continue to shed for up to 6 months. And so this is why Typhoid Mary was such a problem, as you can imagine, is because even if she wasn't currently ill, or maybe she was never ill, even if you do survive this, you are potentially transmitting it to others. So hand hygiene is going to be a major, major thing. Specific symptoms to this, we kind of already mentioned this on the earlier slides, so I'm going to go ahead and progress. As far as epidemiology goes, this does have a reservoir of humans as well as animals in the GI tract. It is transmitted through a fecal-oral route. It is also affiliated with pet reptiles, especially small turtles, less than 10 centimeters, and iguanas, where the carriage rate is about 90%. So I would just always assume that you can love those animals, own those animals, have a great time with those animals. But as soon as you're done, you need to make sure you wash your hands, and definitely don't put any turtles in your mouth. I say that, and you're probably laughing on the other end of this, but this has caused outbreaks, is when especially young kids get a small baby turtle under 10 centimeters and try to give it a kiss and put it in their mouth momentarily, and now they've just seeded themselves with salmonella. It's also affiliated with commercial chicken production. You should always assume that meat and eggs are contaminated. You can get lucky, but I would not gamble with this kind of thing. So always make sure your chicken is cooked to a well done, avoid cross-contamination, that sort of thing. This salmonellosis causes about 1.4 million cases a year. And again, this is likely an underreported number. It totals 400 deaths annually. But luckily, it has a fairly low mortality rate. People usually survive. The deaths, when they're seen, are mostly going to affect infants and the very old, who are already immunocompromised, maybe with weaker GI systems than we would have. So I mentioned for prevention, good hand hygiene, sanitary practices, and avoiding cross-contamination. Those will be really big things. Diagnosis for this includes culture from the stool or from food, and we can also use DNA-based testing like PCR. So typhoid fever, we've done a lot with already, talking about Typhoid Mary, so we'll just hit the highlights here. As you know, the etiology for this is Salmonella typhi, so that's what we're talking about here. Its epidemiology is interesting, though, because this one has no animal involvement at all. It is usually only human-to-human transfer. It is now, thankfully, more of a problem in the developing world, and I don't say thankfully because I want them to suffer, but I'm thankful that it's less of a problem where we live now because of greater sanitation practices. I would suspect that if sanitation practices improve in the developing world, that they would also see a reduction in cases. About 70% of U.S. cases for that reason are acquired during foreign travel. And the incubation period for this can last as long as 2 to 3 weeks, and recovered patients can become chronic carriers. Now, Typhoid Mary Mellon, was a chronic carrier as well, but based on her reporting, she never really had an acute version of the illness. Okay. Sorry. So to recap, typhoid is only going to be transmitted human to human. There's no animal intermediary. But with salmonellosis, you will see the infection moving between humans and animals on occasion as well. So if we look at the preponderance of, or the incidence of typhoid fever and salmonellosis in the United States, you can see that it's really been on the decline since the mid-30s. And it's basically at very, very low levels now in this country. However, salmonellosis has taken a much bigger hold, becoming a really big issue in the 1980s. And now it's sort of leveled out at this rate of about 15 cases per 100,000 of our population. So that's pretty high. The symptoms of typhoid fever, we've talked about this before. Include things like high fever, a continual headache, diarrhea in the second or third week when the fever goes away. And this can progress to ulceration and perforation of the bowel, which can be fatal. The infection is associated with a 20% mortality rate when untreated. And this is probably why we saw so many deaths in the case of typhoid Mary. Pathogenesis for this organism, just like Salmonella discussed earlier, it's going to be phagocytosed, survive phagocytosis, and then divide inside of that cell to avoid the immune system. It will then move into the spleen and liver, and that's where it's going to reside and be part of that chronic carriage. And then it can cross into the bloodstream in addition to cause things like septicemia. Treatment for this, we can use antibiotics, but usually it's going to be if the infection hasn't resolved on its own for a while. The individual will most likely be on antibiotic therapy for weeks at a time. And we've tried to limit this as much as we can because resistance is becoming an increasing problem. As this becomes a more frequent infection, it's being bombarded with antibiotics at more regular intervals. The other problem with this, well, no, that's not really true, so never mind, scratch that. All right, so we'll move on to cholera. We've talked about this before, and now it's time to see what this actually looks like. So the etiology of this is from Vibrio cholerae, which is a curved gram-negative rod with a single polar flagella. The risk factors for contracting this illness include having impaired stomach function and taking antacids. The pathogenesis for this is going to require infection with a very high infectious dose. And it's going to be a fecal-oral ingestion route of transmission. The organism is going to grow in the small intestine and produce an exotoxin. That's one of those AB toxins. So this is another one of those weird examples where an exotoxin is produced by a gram-negative. Usually we associate those with gram-positives, but cholera and Bordetella pertussis are both examples of ones that sort of violate that rule. This toxin is going to cause host cells to secrete water in mass, as well as electrolytes, especially potassium. This massive fluid loss is going to trigger shock, so that dangerously low blood pressure, as well as organ collapse and death. So the symptoms for this are the hallmark feature of this is called rice water stool, and there's kind of a fun looking picture of that over on the right. This really, it doesn't look like normal stool. It's mostly water, first of all. And it kind of has like the appearance of chicken broth. So keep an eye out for that. What you're really seeing is not really stool so much as masses of intestinal mucus and epithelial cells. These people will also have super violent vomiting. They will lose up to 12 to 20 liters, which is 3 to 5 gallons of fluids per day. So you can imagine how this would become rapidly fatal. The mortality rate for this is 50% if left untreated. But less than 1% if we can get them onto oral rehydration and IV rehydration methods as soon as possible. So for diagnosis, it's going to be based on symptoms if we see that rice water stool, as well as a fecal culture. For prevention, you have to maintain really high standards of sanitation, and we do have an oral vaccine that's available. We tend to give it to those that are traveling, for example, to an endemic area, that sort of thing. For the epidemiology of this in the United States, we really only see occasional cases of this, and it tends to be limited to the Gulf Coast area. But as you saw with the Haiti outbreak that happened, they had an earthquake, and that really devastated their sanitation. And so then what ended up happening was peacekeepers came from the United Nations to try to help. And people from peacekeepers from Nepal, which is over here, came to Haiti, right here circled, to try to help. And unfortunately, they brought Vibrio cholerae with them because it was the Nepal version that caused the outbreak over here. So it's one of those situations where, you know, you're trying to do good, you're trying to help people, but because of that poor sanitation situation, it unfortunately resulted in pretty widespread exposure. Treatment for this is going to include rehydration, usually through intravenous methods, because they usually can't keep water down through the GI system, so we kind of have to prop them up intravenously. We also have antibiotics that we can use. This is the reason why, genetically, why cystic fibrosis exists in the human population. So this is a point of evolution. Pathogens are a selective pressure in our evolution. And so cholera, or Vibrio cholerae, the AB toxin, is going to target a potassium chloride channel. And so your bodies or the cells swap potassium and chloride across the membrane to try to drive different solutes and food particles into the cells. So the AB toxin is targeting that. So for if you've ever done a Punnett square sort of situation, you know that there's two versions of any given gene. You get one from mom, one from dad. So you have two copies. So if you have what's called the dominant version, then your potassium chloride channels work just fine. If you have two of those, all the better. If one of those, or not one of those, if both of your copies are what's called the recessive version, that means that those potassium chloride channels don't work very well, or they don't work at all. So the good shot about that is that cholera can't really affect you, right? Because you don't have the right target for them. The bad news is you have cystic fibrosis. So this will mean that you have impaired mucus and fluid absorption in the lungs, as well as in the GI tract. But if you are fortunate enough, I guess, to be the individual that has one functional copy and one non-functional version of the alleles that would be required for that transmembrane system, then you are immune to cholera, because only 50% of your cells will have, or you'll have 50% as many of those receptors that the toxin is going to target. And so that will be just under the threshold of what's required for really serious disease. So that's a long-winded explanation, but that's why cholera exists, or that's why cystic fibrosis exists. So E. coli gastroenteritis, we could spend an entire semester talking about this. But E. coli, it's really interesting, right? Because it's a normal flora organism of the gut, and it's present in huge quantities. But if you're having diarrhea and you think it's E. coli, how in the world do you differentiate between the ones that are supposed to be there versus the ones that are causing an actual illness? And so for that, we use a special kind of media. We've worked with McConkey agar in lab. This one is called sorbitol McConkey agar, or SMAC. So it's a little bit different, because it's going to, much like the McConkey plate, only gram negatives can grow on this. And we can differentiate regular run-of-the-mill E. coli that's found in your intestine based on its ability to ferment sorbitol. And then for E. coli 0157H7, for example, it will not appear pink. And so that's how you would tell the difference. There are 5 different pathogenic varieties of E. coli that can cause illness. They have different abbreviations, and they stand for these things here. We'll go through each one briefly. So for enteropathogenic E. coli, which is EPEC, this is a major concern in developing countries where it is fatal in infants. In this situation, bacteria will attach to the intestinal wall, and they'll eliminate those surrounding microvilli and cause the host cell to form these pedestal structures beneath the organism to sort of better facilitate this transmembrane interaction. So this is one of the interesting features of this. For enteroinvasive E. coli, this is going to have, as the name might imply, it's going to invade and affect the blood and lymph. So it's going to be nearly, or not blood and lymph, but it's going to invade, it's going to kill some of those epithelial cells and dig a little bit deeper. It's nearly identical to Shigella in terms of its pathogenesis, and it will include things like inflammation, fever, and a Shigella-like dysentery. Enteroaggregative E. coli, which is what EAEC stands for, is only found in humans. It doesn't have an animal reservoir, and it tends to form what looks like a stacked brick configuration when you look at it in tissue culture. It's not generally invasive, but it does produce an enterotoxin that produces a watery diarrhea. One of the most important ones, though, is EHEC, which stands for enterohemorrhagic E. coli. This is also known as Shigella-like toxin-producing E. coli, or STEC. This is the only pathogenic E. coli that produces this toxin, and it includes probably the most famous example of O157H7, which has been associated with ground beef outbreaks, spinach outbreaks, you name it. Its pathogenesis is interesting because it doesn't actually invade the intestinal wall. It releases that toxin into the lumen of the intestine. The toxin is then released upon cell death, so we don't necessarily want to give antibiotics in this case because it will just worsen the disease. The main reservoir for this is cattle. About 2 to 3% of them carry it asymptomatically. So this can be transmitted by flies, for example, that land in the cow dung and then fly to your kitchen table and land in your spaghetti, for example. Otherwise, and this is really the bigger cause here, is that you'll get contamination of the carcass during slaughter and potentially the contamination of other parts of the beef. And so this is why, especially things like ground meat is so risky because it's not just coming from one animal. It's kind of the spare bits that weren't cut up into steak and loin and roasts, all mixed up into one thing from hundreds of animals. And so if just one of them happened to have E. coli in it, then now that's contaminated the entire batch. And so you can imagine as those meat products are distributed over a wide geographic range, that it can really hurt a lot of people. We do have a new vaccine for use in cattle, but it's a little bit too early to see if this is really helping yet. But we have good hopes. Transmission for this is going to be contaminated food or water. An additional route is through farms and petting zoos. So anytime you go to something like that, you should really be very careful to wash your hands after touching, feeding animals, playing in the dirt of one of those locations, that sort of thing. And so one of the cases that's in your differential diagnosis, we'll be concerned with this. But risky sort of things include ground meats, as I mentioned before, and then leafy vegetables. And so this really is interesting because the reason why is because a lot of these farms that grow produce are downstream of feedlots. And so really it's runoff irrigation from the feedlots that then feed into these rivers and streams that farmers then use to water their lettuce or their spinach, that sort of thing. And since those produce items are not cooked before you eat them, and they're actually kind of hard to wash apart from just water, the organism can really set up shop there. Unfortunately, this one has a very low infectious dose of about 100 bacteria, so it really doesn't take much. And the results can be rather devastating. Most of the patients will have a self-limiting diarrhea, so it might look like anything else. But a proportion of them will take on the additional symptoms of a hemorrhagic colitis, so a bloody diarrhea, and also hemolytic uremic syndrome, or HUS. This is when you have blood in the urine, often leading to kidney failure. And survivors of this will often require kidney dialysis or transplant. It affects about 5 to 10% of children who are infected with this organism, with a mortality rate of about 5%, causing 200 to 500 deaths annually. For treatment, we use IV rehydration and monitor serum electrolytes as well. This organism came to prominence really and became a household name during the 1993 Jack in the Box outbreak. And some of you weren't even alive for this yet, but it was a major event in food safety history. This was an outbreak that affected Western states, including Washington, Nevada, California, and Idaho. I actually grew up in Washington while this was happening. I was 12 years old. So this was happening to kids in our community. It was a really scary time. But anyway, the outbreak caused 732 cases, and it was from contaminated beef products or beef patties at Jack in the Box that weren't fully cooked. They were running this special about this monster burger, and it was in high demand. People were coming in and wanted to eat it. And as you know, if any of you have worked in a restaurant, and I know many of you do, when things are happening fast, sometimes corners get cut. And so they weren't cooking the patties as long as they probably needed to. These patties were probably being served more along the lines of medium or medium rare. And so it was not killing the organism. This illness disproportionately affected kids under the age of 10, resulting in 4 deaths. All were in children. And so this led to a major sort of come to Jesus moment by our government to be like, we had 732 cases and we didn't even know what was going on until it was too late for a lot of people. And so now we've set up this thing called PulseNet. And I actually used to work in this lab at CDC in Atlanta. But what it is a network of every public health lab in the country, state-wise anyway, and some of our locals too. And they all, because Salmonella and E. coli 0157H7 are mandatory notifiable diseases, these organisms will be referred to those public health labs. So then the public health lab will do DNA fingerprinting on them. It's called pulse field gel electrophoresis, or PFGE. It comes up with this banding pattern. And in that way, we can see how related these illnesses are. So if we start to see the same banding pattern, there isn't an example here where they're all the same, but let's say like this one right here looks very similar to this one. And if we start to see a lot of this pattern, it might indicate a common source of infection or a common reservoir. Maybe we'll end up finding out that it's from chicken wings, or it's from tomatoes, or something of that nature. And so anyway, these public health labs have to upload their patterns to this national database. And then database managers at CDC will take a look at what's going on across the entire country within that organism. So one of these people is assigned to 0157H7 and others assigned to Salmonella, et cetera. And so they work with epidemiologists and combine this data with what we know about cases that are occurring and where to try to get down to that information. And so if PulseNet had existed when the Jack in the Box outbreak happened, instead of being 732 cases, it probably would have been closer to like 70. We could have dropped it by about tenfold because we would have been looking for it. And so this is a major thing here. It's really revolutionized food safety. And now, actually, E. coli 0157H7 has been declared an adulterant by the FDA. So this is the same sort of idea as if glass had gotten into the food supply, or cockroach pieces, or whatever. If that's there, they have to do a mandatory recall. And this was a big deal because up until this time, they didn't think that bacteria could be an adulterant because it was just a natural part of the process, and that's a natural thing that's found on the animal during slaughter. But they decided that this was so dangerous that it really should be declared an adulterant, because no amount is safe. When only 100 cells can kill you, then really no amount is safe. So the next one is enterotoxigenic E. coli. This is diarrhea due to enterotoxins that will resemble the cholera toxin, which is an AB toxin. And the organism itself will remain in the intestinal lumen. This is frequently fatal in children under 5 years old, especially in the developing world. And this is the most common cause of what's called traveler's diarrhea. So when you go to another country, you drink the water there, or you eat produce that you didn't handle yourself, then you can get those illnesses. For treatment, it's going to include oral rehydration and antibiotics in severe cases. We wouldn't want to do this every time because of this toxin that would be released. So prevention: when you travel, only drink bottled water and avoid produce that can't be washed or peeled or cooked before you eat it. So even though the pineapples are delicious in Central America, unless you cut and washed that yourself, then you need to not be eating it. Okay. Campylobacter gastroenteritis is another interesting one. Very common cause. In fact, it's the leading cause of foodborne illness in the United States. While those other items were splashier titles, this is the one that is actually a lot more prevalent. So these are gram-negative microaerophiles. So they do okay in the intestinal environment, and they're spiral shaped. This organism is especially well adapted to the chicken intestine. It seems to really like that environment. So you should assume that all chicken is contaminated. All parts of the chicken is contaminated. Cattle will even excrete it in their feces and milk. But we haven't seen this as a route of infection necessarily. This is almost always associated with chicken. For symptoms, it's going to include a fever, abdominal pain, and diarrhea, but not necessarily vomiting. The treatment for this is self-limited. It'll usually resolve within a week, but there can be complications. And in this one in particular, About 1 in 1,000 cases will progress to Guillain-Barré syndrome, which is a temporary autoimmune-driven paralysis. What happens here is there's molecular mimicry where the organism's surface molecules look like a lipid component of the nervous tissue. So you clear the infection, your immune system has developed antibodies against Campylobacter, but because those things that the antibodies are directed against are so similar to nervous tissue, those antibodies are going to start attacking, and that will be pretty bad. So Clostridium difficile associated diarrhea, we've talked about this before. We're going to give it a little bit more depth here. It's a gram-positive endospore-forming anaerobe. Its pathogenesis is mainly driven by the production of an exotoxin. And its symptoms are going to range from like a mild diarrhea to a life-threatening colitis, which can include things like ulceration and perforation of the bowel. Its risk factor or at-risk populations will be those who have had extended use of antibiotics, especially the fluoroquinolones. That would include things like Cipro and Levaquin. In the clinical environment, those are their trade names. It is considered, for that reason, a healthcare-associated infection. It's also seen in daycares. And actually, this is becoming sort of one of the most prominent healthcare-associated infections we have. Its highest mortality is observed in the elderly, especially as they require additional medical intervention. Next, we're going to move into the viral infections. And this will include things like mumps, which we don't see very often anymore because of a vaccine. All the ways you can get hepatitis, viral gastroenteritis, and so we'll talk about all of those. What's interesting about viral infections is that they don't replicate within the contents of the digestive system. So they won't replicate inside the lumen of the intestine or the stomach or any of those tissues. They have to invade some of the accessory organs like the liver, the gallbladder, the pancreas, the parotid glands in order to cause illness. So first we'll talk about mumps. Again, this is a pretty rare infection. We don't see this very much anymore. Its target tissue is the parotid gland. So if we look at the head structure here, we've got the oral cavity, the sinuses up here. And right back here, right underneath your ear, is the parotid or salivary gland. And this is what mumps is going to be looking for. It's going to show up symptom-wise 16 to 18 days post-exposure. So it might be a hard thing to link back if it were more widespread. The symptoms are going to include a painful swelling of one or both glands. So with this man, you'll see that he has a character or a shape on the right side or his left side, and you see that it's extended, distended right here, and it doesn't really make sense. And so you'll see this really big neck sort of thing going on. You'll also see fever and pain when swallowing. In men, this can also lead to sterility as the testes can become inflamed. The virus can also cause complications including meningitis, inflammation of the ovaries, and pancreatitis. Its transmission is through saliva or respiratory droplets, and you're most contagious within the first 48 hours after symptom onset. Outbreaks of this are very rare in the United States because we have prevention using the MMR vaccine, which stands for measles, mumps, and rubella. So we'll move on to hepatitis, which is an inflammation of the liver. So on the top, we have what looks like a healthy liver. And on the bottom, you can see the effects caused by hepatitis C. So very drastic effects. They're going to be caused by several unrelated viruses. And there can also be contributions from things like alcohol use and drug use. So hepatitis, there are 5 different variants of this, and we'll go through each of them. All of them are caused by different viruses. So hepatitis A is caused by the hepatitis A virus, which is a single-stranded RNA virus. You'll notice this is going to change throughout. Its transmission is fecal-oral, and oysters can be contaminated, so that's a significant vehicle. There are no other animal reservoirs. It tends to be shed in the feces and is detectable in blood and urine. Its incubation time is about 4 weeks. The highest viral count observed in the bloodstream, though, is going to be just before symptoms appear. So it can sometimes make it hard to diagnose if those serum titers are not very high when you're actually exhibiting symptoms. For pathogenesis, this virus is going to replicate in the epithelial lining of the GI tract, then advance to the bloodstream to cause viremia, and then target the liver, kidneys, and spleen. Symptoms for this will last 2 to 21 days with very low mortality. In fact, many of these are going to be subclinical. People won't know that they're infected. But for those that do display symptoms, it'll include things like anorexia, where they're dropping weight for no real explainable reason, except for their other symptoms that include nausea and diarrhea. Malaise is going to be an issue, abdominal discomfort, as it affects things like the liver, fever, chills, jaundice, dark urine, and the liver itself will be tender and enlarged. So you might see some distension of the belly as well. So risk factors for this illness include low socioeconomic status. This has to do with education and access to healthcare. Also injecting drug users, not necessarily IV, it would be injecting drug users who share needles. And also there seems to be a preponderance in homosexual men who are sexually active. Diagnosis for this is going to depend on serology, so we'll be looking for the presence of antibodies or virus in your bloodstream. Treatment for this, unfortunately, there is none for active disease, but we can give immunoglobulin to exposed contacts to try to bind up that virus before it can affect them. So this is really not going to help anybody that's already showing symptoms, but might prevent the spread. For prevention, thankfully, we have a vaccine for this, and we now administer this during the childhood series. It's also available for you as an adult and for high-risk groups like healthcare workers or those traveling to endemic areas. I would imagine all of you have been vaccinated for this, or if not, you will be before you start doing any clinical rotations. Okay, hepatitis B. Also kind of has a name that tells you what it does, hepatitis B virus. This is a double-stranded DNA virus with an envelope. Its transmission is through body fluids. So this one can be transmitted through blood, saliva, breast milk, semen. It can also be from mother to baby during the process of birth. Risk factors for this include being a healthcare worker, so get your vaccine, and then also IV drug users. So for this, chronic carriers are going to be more common in certain individuals, and this is really interesting, especially people from Asia, Africa, and the Mediterranean. There's a typo there, and I apologize. For prevention, we have a hepatitis B immunoglobulin to newborns that we can give, and then there are two forms of this. You can have an acute and a chronic infection. So for acute, this can be subclinical, but if you do have symptoms, you'll have things like a low-grade fever, nausea, abdominal pain, jaundice, and dark urine, kind of like what we saw for hepatitis A. But the complications include sudden massive liver damage requiring a liver transplant. For chronic infections, though, this will be when the acute situation, which we just described, lasts more than 6 months, which sounds like a pretty awful situation. This will mostly occur in the young. Those individuals will have malaise, sort of just feeling yucky, a loss of appetite, fatigue, usually no jaundice, but it can progress to cirrhosis or liver cancer. So for this, you're going to, I think we had a prevention thing up here. We did. We've got the H-big for newborns, but we can also prevent by encouraging the use of disposable needles and syringes, barrier type contraception during sexual encounters. So oral contraception is not enough. You need to be using a condom or spermicide or some other sort of thing. There's also a vaccine available for this in the childhood series that we give routinely now, and I believe you can still get this as an adult. We also do this thing now where we screen blood before we include it in the blood bank. And this actually comes to us from an African-American physician named Charles Drew. We don't have very many African-American heroes of science because they've historically been left out of things. They've been discouraged from becoming scientists and doctors. So this is kind of a great thing to see. But Dr. Drew was an American physician. He lived from 1904 to 1950, and he really developed the blood banking techniques that we have now that screen for a lot of these pathogens to prevent, prevent a lot of those difficulties. So diagnosis for this is going to include things like liver and liver tests, symptoms themselves, and serology. And for treatment of this, you're going to be doing supportive therapy, so treat the symptoms for acute infections, and then use antivirals or liver transplant, for those who are chronically infected. Hepatitis C is caused by the hepatitis C virus. It is a single-stranded RNA virus with an envelope, and its transmission is going to be due to things like blood transfusions, sexual contact, and those who are, again, injection drug users who share needles. Pathogenesis is going to, or how it causes its disease, is through triggering an inflammatory response that either clears the infection or slowly destroys the liver. It can mutate rapidly to avoid the immune system, which makes it an additionally complicated situation. It can take, unfortunately with this, 20 years for symptoms to appear, and 85% will progress to chronic hepatitis, which will then contribute to things like cirrhosis of the liver or even requiring liver transplantation. As far as prevention goes, you want to make sure you don't share personal use items, even things like, you don't even want to share things like razors or anything of that nature. There is unfortunately no vaccine available, so you would want to take prevention pretty seriously with this. Treatment, we can use long-term antivirals, but they're very expensive with severe side effects, so not ideal. Then we're going to round things out with two of our more minor hepatitis versions. Hepatitis D can occur as an acute or chronic infection. It tends to be linked to the epidemiology of HBV, or hepatitis B virus. It is most often seen in injection drug users. And then lastly, hepatitis E is going to be differentiated from many of these because it is transmitted through the fecal-oral route. It is endemic in areas with poor sanitation, to include areas like India and Southeast Asia, where it's most common. It can cause a death in 20% of pregnant women, so pretty significant infection. Okay, next we'll talk about rotavirus. This is actually the most common cause of viral gastroenteritis, especially among young kids. Let's see, that's what I needed. And this epidemiology for this, like many of our infections today, are through a fecal-oral route. It has a 2 to 3 day incubation period before symptoms. It is most often seen in winter, and it has, unfortunately, a very low infectious dose, so it really doesn't take much. Wash those hands. Let's see. Symptoms of this are going to include a low-grade fever, diarrhea, and vomiting that lasts for a week. Diagnosis for this will be based on serology, and you can treat this with oral rehydration therapy. There is no vaccine there is a vaccine for this, and we will especially target the immunocompromised babies that were born with congenital defects or have been born premature, who will be more susceptible to the complications of any of these diseases. Okay, this is one that I think almost everybody has had at some point. It's really common. Norovirus, which is also known as Norwalk virus, it's the same thing, is a fecal-oral transmission route, but you also get it from aerosols from vomiting. So they vomit. Some of those respiratory droplets in that process will float in the air. You then breathe it in, and there you go. It has a very low infectious dose. In fact, about 10 virions is all that it takes. And individuals who recover will continue to shed it. So there's this is as far as evolution of viruses go, this is a good one because it knows full well how to get to the next host. It's very common. This is known as the cruise ship virus. And so it'll affect the entire ship basically. And so they'll be really careful about fomite cleaning, handrails, doorknobs, light switches, that sort of thing. But it's also been associated with outbreaks at restaurants too. It accounts for about 50% of foodborne viral gastroenteritis, and its incubation period is, about 18 to 48 hours. My husband got it once from a casino, so I would not be very careful about washing your hands when you go into a high-risk environment like that. For diagnosis, this would be based on DNA testing or PCR, as well as serology of, it says serology of stool. Oh, so that what that means is that we're using a antibody where we know full well what it binds to. We test that against a stool suspension, and if that antibody finds its target, then we'll be able to see that. Symptoms for this are going to include vomiting with or without diarrhea for about 2 to 3 days, and it can be significant. Treatment for this is going to include rehydration, both orally and intravenously, in serious cases. We already talked about rotavirus. There we go. Next, we'll move on to the protozoan infections. Giardiasis is caused, or Giardia is more commonly named, is caused by Giardia intestinalis. This used to be called Giardia lamblia, which was my favorite, but they've changed the name. This organism, you can see it here, this is called the trophozoite. It has these, it looks a little bit like a stingray, and it's got flagella that go with it, trailing off. And so this will attach to the intestinal wall, causing symptoms of malaise, nausea, flatulence, or farting is the common vernacular, weakness, weight loss, and abdominal cramping. It's most often seen during camping and swimming seasons. It does inhabit recreational water sources like lakes and streams and that sort of thing. And campers will sometimes use stream or lake water for their hydration. So we have iodine tablets that you should use to reduce the possibility of a Giardia transfer. So transmission, as you might have guessed at this point, is through contaminated water supplies, so an ingestion or vehicle transmission. The diagnosis for this is the string test, and this says EU to remind me to tell you what that is. So what happens is you take a gelatin capsule like you would normally have like antibiotics in, and they basically tie a string to that. They tie that string end to the side of your cheek on the outside of your mouth, and then you swallow the capsule. That capsule goes down through your stomach, through your small intestine, and once it gets there, so they let this sit with this string hanging out of your mouth for probably like a good 8 hours. Then they pull that up. So we're pulling up through sphincters that shouldn't normally open in a reverse fashion. We're doing that, and basically what was in your intestine is now in your mouth as they pull that out. And then they look at the fluid that's trapped inside of that capsule, and they can see things like the trophozoite or the cyst in that suspension. So that's how they diagnose. Treatment for this is going to be oral medications that are good for protozoa. You can also have amoebic dysentery, which is caused by amoeba. This is most often caused by the organism Entamoeba histolytica. Again, contaminated food or water is the risk here. Cysts for this organism are going to be resistant to the stomach acids. So they will get to the intestine and mature to trophozoites in that tissue. And there they will feed on the tissue of the intestine. So if this is normal mucosa over here, you'll see this ulcer, and it kind of looks like an Erlenmeyer flask like we use in the laboratory sometimes. And so they're called flask-shaped ulcers. Severe dysentery is associated with this, including things, or it is a severe dysentery, including blood and mucus-filled feces. You can have secondary infections by bacteria during this time. As you can imagine, if we're disrupting this mucosal barrier, then anything can get in here and access this tissue. Diagnosis from this is going to require finding the pathogen in the stool, so maybe a fecal smear. And then you can treat with oral medications like metronidazole and iodoquinol, We're going to round things out by hitting the highlights of our helminthic infections. These are things that are caused by animal parasites. So ascariasis, we've already heard about from one of our disease presentations, but it's caused by the organism Ascaris lumbricoides. This is most common in the American Southeast where we live, affecting 20 to 60% of children, maybe closer to the 20% now with modern awareness. People wear shoes, for example. The shoe thing is more of a hookworm problem. But well, anyway, it's less, hopefully closer to 20% now. If we look at all of the infections that are caused by animal parasites in the United States, ascariasis, is really the leader here, followed by hookworms, enterobius, and some of our other worms here. So diagnosis of ascariasis is going to be because you'll actually see the worms coming out of the anus, mouth, or nose. And this is what, oh no, that's a hookworm. A hookworm is this animal is called Necator americanus. It is, again, most common in the American Southeast, where I guess sort of a hotbed of parasites. So symptoms for this are going to include anemia, lethargy, and pica, which is a phenomenon where you want to eat things that are not food. So things like clay or other hair, other weird things that are not food and not really appetizing in any way. The prevention for this is to wear shoes in soil. And so this is becoming less of a problem. I was trying to say this earlier because we've become, I guess, a more civilized society, and people tend to wear shoes more often, especially when they're outside. But that's how you would get that. Through little breaks in your sole of your foot, things would be able to get in. And so it's a hookworm, its mouth is right here, and on it has these hooks that it can use to grab on. Pinworms will round this out, is the organism Enterobius vermicularis, its symptoms is basically itchy anus where the eggs are laid. So the female will come out of the anus, lay their eggs right there. And those eggs, as they're germinating and hatching, are really what's causing inflammation there. So that's really what's going to happen here to diagnose is the physician is going to take kind of like a piece of Scotch tape, apply it to the anus, take it off, and then look at that under the microscope, and there they will see eggs. So a very fun time. So there's a review slide here, a chart for you to go through and see if you can sort of remember or work through the different viruses that can cause gastrointestinal illness. And so I would encourage you to work through that. Also, in your textbook, I would recommend looking through these questions. They're in the back of the chapter. If you have questions or need answers for any of those, please let me know. And then, of course, there's the differential diagnosis slides that accompany this lecture and should be available through Brightspace. Those are always a really productive exercise, and I hope you saw that on the exam. So with that, I am going to go ahead and close. Have a great day, and I'll see you soon.